Every human life begins with a story that sounds simple. A sperm meets an egg. That is the version most people remember from school. Clean, direct, almost gentle. But inside the human body, the real process looks nothing like that. Because fertilization is not a meeting. It is a survival test. And almost every cell that begins the journey will fail. In a typical ejaculation, the male body releases somewhere between 200 and 300 million sperm cells. At first glance, that number seems excessive. But biology rarely wastes energy without reason. The vast majority of these cells are never meant to succeed. They exist to be filtered out. Because the female reproductive system is not a passive environment waiting for fertilization. It is one of the most selective biological landscapes in the human body. The first obstacle appears almost immediately. The vaginal environment is naturally acidic. This acidity acts as a defense system, protecting the body from harmful bacteria and pathogens. But sperm cells are extremely sensitive to this chemical environment. Within minutes, millions of them lose mobility. Many die before they even reach the cervix. The race has barely begun, and the numbers are already collapsing. Those that survive the first barrier approach the cervix. But the cervix is not simply an open doorway. It produces thick cervical mucus designed to control what enters the uterus. Most of the time, this mucus is dense and restrictive. Only during a short window near ovulation does it become thinner, creating microscopic channels that sperm may pass through. Even then, the passage is narrow and selective. Only the strongest swimmers manage to move forward. Millions more are left behind. If a sperm cell successfully crosses the cervix, it enters the uterus. The journey is far from over. The uterus is not empty. It contains immune cells that constantly patrol the environment, searching for foreign invaders. To the immune system, sperm are exactly that. Foreign. Many sperm are destroyed here by the body's own defenses. The numbers shrink again. What began as hundreds of millions is now reduced to only a few thousand. But the final destination lies even farther ahead. Beyond the uterus are the fallopian tubes. These thin passages connect the uterus to the ovaries. Somewhere inside one of these tubes, a single egg cell may be waiting. But the tubes are narrow. Fluid currents push against the sperm. Muscular contractions alter the path. Many sperm become trapped. Others simply lose the energy required to continue. By the time the leading sperm approach the egg, only a few hundred remain, out of the original hundreds of millions. And then they encounter the egg itself. The egg is the largest cell in the human body, almost unimaginably larger than the sperm approaching it. But size is not its only defense. The egg is protected by multiple biological barriers. The first is a layer of supportive cells called the corona radiata. These cells surround the egg like a protective cloud. Sperm must push through this layer before reaching the egg's outer shell. Beneath it lies something even more formidable, a glycoprotein barrier known as the zona pellucida. This shell is not simply structural protection. It acts like a molecular lock. Only sperm carrying the correct biochemical signals are allowed to bind to its surface. The rest are rejected. When a sperm successfully attaches to the zona pellucida, a remarkable transformation occurs. At the tip of the sperm head is a structure called the acrosome. Inside it are powerful enzymes. Once binding occurs, the acrosome releases these enzymes in a process known as the acrosome reaction. These enzymes begin dissolving a microscopic pathway through the outer shell of the egg. But even this is not guaranteed success. Many sperm attempt this process. Most fail. Their energy runs out before they can reach the inner membrane. Only one will go far enough. Eventually, a single sperm breaches the egg's outer membrane. At that moment, the egg responds instantly. Chemical signals trigger a defensive reaction across the entire surface. The zona pellucida hardens. Its structure changes. Every other sperm is permanently locked out. The race is over. All remaining competitors are rejected. Inside the egg, the victorious sperm begins to transform. Its tail dissolves. Its nucleus expands. The genetic material carried from the father reorganizes itself into a structure called the male pronucleus. At the same time, the egg prepares its own genetic material. This forms the female pronucleus. These two structures slowly move toward each other through the cytoplasm of the egg. Each one contains half the genetic blueprint required to build a human being. 23 chromosomes from the father, 23 chromosomes from the mother. When they meet, the membranes dissolve. The chromosomes align. And for the first time, a complete human genome exists. 46 chromosomes combined into a single cell. This cell has a name, the zygote. In that microscopic moment, something extraordinary has happened. Not just fertilization, but the creation of a completely unique genetic identity. A blueprint that has never existed before and will never exist again. Eye color, height potential, metabolism, brain development, millions of traits encoded in molecular sequences of DNA, all contained inside a cell smaller than the tip of a needle. But the most remarkable detail is not its size. It is what that single cell now knows how to do. Because hidden inside the zygote is the complete biological instruction set required to build an entire human body. 200 different cell types, trillions of individual cells, organs, tissues, and neural networks of extraordinary complexity, all beginning from one microscopic starting point. Life did not begin with certainty. It began with elimination. Hundreds of millions entered the race. Almost all were filtered away, and only one cell remained. Not because the process was easy, but because biology was designed to be brutally selective. Every human life begins the same way, not with abundance, but with survival. At the moment fertilization is complete, the newly formed zygote contains everything required to build a human being. Every instruction, every structural plan, every biological program, all compressed into a single microscopic cell. But possessing the blueprint is only the beginning. The real challenge is turning that blueprint into structure. And the first step happens almost immediately. Within hours, the zygote begins to divide. One cell becomes 2, then 2 become 4, 4 become 8. This process is known as cleavage. Unlike normal cell growth, these divisions happen rapidly and without increasing the overall size of the embryo. Instead of growing larger, the single original cell is being partitioned into smaller and smaller units, each one carrying the same genetic instructions. The embryo remains roughly the same size it was when fertilization occurred. But inside, complexity is beginning to multiply. After several rounds of division, the embryo reaches a stage known as the morula. At this point, it contains around 16 to 32 tightly packed cells. Viewed under a microscope, it resembles a tiny cluster of berries. This cluster is still traveling, because fertilization did not occur inside the uterus. It occurred higher up, inside the fallopian tube, and the embryo is slowly drifting downward toward the uterine cavity. Tiny hair-like structures lining the fallopian tube, called cilia, gently push it along. Muscular contractions assist the movement. The journey takes several days. During that time, the embryo continues dividing. Not growing, dividing. Soon the structure changes again. Fluid begins to collect inside the mass of cells. A hollow cavity forms. The embryo now enters a new stage called the blastocyst. And something crucial happens here. For the first time, the cells begin separating into different roles. One group gathers on the inside. These cells form what is called the inner cell mass. They will eventually develop into the entire human body. Every organ, every tissue, every neuron in the brain. The second group forms the outer shell of the blastocyst. These are the trophoblast cells. Their future is different. Instead of forming the body itself, they will build the life support system that keeps the embryo alive. They will form the placenta, the structure that connects the developing fetus to the mother's body. In other words, even at this extremely early stage, the embryo is already organizing labor. Some cells will become the organism. Others will build the environment that allows it to survive. But the embryo is still trapped inside something left over from the egg. A protective shell known as the zona pellucida. This shell was essential earlier. It prevented multiple sperm from entering the egg. Now it has become a limitation, because the embryo cannot attach to the uterus while it remains sealed inside this layer. So the blastocyst performs another remarkable maneuver. It begins to break free. Through subtle pressure and chemical signaling, the embryo slowly pushes against the shell. Eventually, a small opening forms. The blastocyst squeezes through, cell by cell, until it finally emerges. This moment is called hatching, and without it, pregnancy cannot continue. Now completely exposed, the blastocyst drifts into the uterine cavity. The lining of the uterus, known as the endometrium, has been preparing for this moment. During the menstrual cycle, hormones thicken this tissue and increase its blood supply. The surface becomes soft and nutrient-rich. But the embryo cannot simply rest on top of it. To survive, it must attach. And then it must do something far more aggressive. It must invade. The outer trophoblast cells begin to interact with the uterine lining. They release enzymes that allow them to break through the surface layer of the endometrium. Slowly, the embryo embeds itself deeper into the tissue, almost like a seed pushing roots into soil. But biologically, the process is more intense than that comparison suggests. The trophoblast cells actively burrow into the uterine wall. They reshape surrounding blood vessels. They establish the earliest connections that will eventually become the placenta. From a purely biological perspective, the behavior resembles something almost parasitic. The embryo is restructuring the mother's tissue in order to build its own support system. And the mother's body allows it. Because hormonal signals from the embryo are already communicating with the maternal immune system. Instead of rejecting the embryo as a foreign structure, the body begins to cooperate with it. But this stage is incredibly fragile. A large percentage of fertilized eggs never successfully implant. If implantation fails, the embryo cannot access nutrients or oxygen. Without those resources, development stops. The process ends before pregnancy is even detected. Most people never realize it happened. In reality, the earliest stage of human life contains an enormous amount of biological uncertainty. Even after fertilization, even after several days of development, survival is still not guaranteed. For the embryos that do succeed, however, a critical milestone has been reached. The blastocyst is now embedded within the uterine wall. Nutrients begin flowing from maternal tissue. Cells continue dividing. Signals begin coordinating more complex patterns of development. The tiny cluster of cells is no longer drifting through the reproductive tract. It has found a place to stay, a place where the next phase of human development can begin. Before organs exist, before limbs form, before the brain begins to take shape, life must first accomplish something far more basic. It must secure its position inside the body that will sustain it. Because before a human can grow, before a body can form, life must first solve a simpler problem. It must find somewhere to attach. Once the embryo has successfully implanted into the uterine wall, something extraordinary begins to unfold. Up until this point, development has mostly been about survival. Reaching the egg, forming a zygote, dividing into clusters of cells, finding a place to attach. But now a completely different phase begins. Because the embryo is no longer just multiplying, it is beginning to organize. And the way the human body organizes itself from this tiny cluster of cells is one of the most remarkable processes in biology. At first, the embryo still looks deceptively simple, just a small disk of cells embedded within the uterine lining. But inside this structure, the cells are starting to receive chemical signals that guide their future roles. Cells begin migrating, shifting positions, forming patterns. This stage is known as gastrulation, and it represents one of the most important turning points in human development. Because during gastrulation, the embryo establishes the three fundamental layers that will eventually construct the entire body. These are called the germ layers. The first layer is the ectoderm. This outer layer will eventually form the structures that interact with the outside world: skin, hair, nails. But even more importantly, the ectoderm gives rise to the entire nervous system, the brain, the spinal cord, every neuron that will one day allow the body to think, remember, and perceive reality. All of it begins within this thin layer of early embryonic cells. Beneath it lies the mesoderm. This middle layer forms the structural systems of the body: muscles, bones, blood vessels, the heart, the kidneys. Much of the body's internal architecture originates here. It provides strength, movement, and circulation. Without the mesoderm, the body would have no framework. The deepest layer is the endoderm. This inner layer forms many of the organs responsible for sustaining life from within: the lungs, the liver, the pancreas, the digestive tract. These organs manage energy, nutrients, and oxygen. They keep the internal environment stable. Even though they are hidden deep inside the body, their foundations are laid at this incredibly early stage. What makes gastrulation remarkable is that these layers emerge from cells that were previously identical. Just days earlier, Every cell in the embryo contained the same potential. Now they are beginning to specialize. Chemical signals activate certain genes while silencing others. Some cells commit to becoming nerve tissue. Others move toward muscle or bone. Still others begin shaping the organs that will process food and oxygen. The blueprint contained within the original zygote is now being interpreted, and the instructions are astonishingly precise. Soon after gastrulation begins another critical transformation. The ectoderm folds inward along the center of the embryo. This folding creates a structure called the neural tube. This process is known as neurulation. The neural tube will eventually develop into the brain and spinal cord. In other words, the very structure that will one day allow a human being to think about their own existence is already beginning to form. And it happens long before the embryo resembles a recognizable human shape. As the weeks pass, development accelerates. Cells multiply rapidly. Tissues expand. Structures that will become organs begin to appear. Around the 4th week, the embryo measures only a few millimeters in length. Yet inside this tiny body, the early heart has already formed, and it begins to beat. At first, the rhythm is simple and uncoordinated, but the movement is unmistakable. A circulation system is starting to emerge. Blood begins to move through primitive vessels. Oxygen and nutrients begin reaching developing tissues more efficiently. The embryo is no longer relying solely on diffusion between cells. It is beginning to build its own internal infrastructure. Meanwhile, the trophoblast cells that invaded the uterine wall earlier continue expanding their work. They are constructing the placenta. This organ is one of the most complex biological interfaces in nature. It connects the developing fetus to the mother's blood supply. But the two circulatory systems never actually mix. Instead, oxygen and nutrients diffuse across microscopic barriers. Carbon dioxide and metabolic waste travel in the opposite direction. Through this exchange system, the fetus receives everything it needs to grow: energy, oxygen, building materials for tissues and organs. The placenta functions as lungs, kidneys, digestive system, and immune interface all at once. And it does this silently for the entire duration of pregnancy. Over the following weeks, the embryo gradually begins to resemble a human form. Small protrusions appear that will become arms and legs. The head grows larger as the developing brain expands. Facial structures begin forming. The eyes shift toward their final position. The heart grows stronger. Neural connections multiply rapidly. By the end of the 1st trimester, the organism is now referred to as a fetus. Many of the major organs are already established. What remains is growth, refinement, and increasing complexity. Inside the uterus, development continues for months. Bones harden, muscles strengthen. Neural circuits form connections that will eventually control movement and sensation. The fetus begins to move. At first, these movements are subtle, often too gentle for the mother to feel, but eventually they become stronger. By the middle of pregnancy, motion inside the womb becomes noticeable, evidence that the nervous system and muscles are coordinating their activity. Throughout this entire process, the original blueprint from the zygote continues guiding development. Trillions of cells are produced, yet each one still carries the same genetic instructions that were formed during fertilization. Different genes activate in different tissues. This selective activation allows the same genome to produce hundreds of specialized cell types. Neurons, blood cells, muscle fibers, skin cells. Every tissue emerges from the same original genetic code. The diversity of the human body arises not from different DNA, but from how that DNA is used. Eventually, after approximately 38 weeks of development, the fetus is fully prepared to survive outside the uterus. The lungs are mature enough to breathe air. The brain has formed the basic structures needed to regulate heartbeat, breathing, and temperature. The muscles are strong enough to move independently. At this point, birth can occur, and the organism that began as a single microscopic cell now exists as a complete human body. But the most astonishing part of this entire process is how it started. Not with organs, not with tissues, not with a brain, only with a single fertilized cell. A cell carrying a set of molecular instructions. Instructions capable of organizing matter into something vastly more complex than itself. Every heartbeat, every thought, Every movement a human will ever make traces back to that original starting point. The human body is not assembled piece by piece like a machine. It unfolds, gradually, systematically, following biological instructions written long before birth. And it all begins with one microscopic cell that already contains the knowledge required to build an entire human life.